So what I'm going to do is go through cellular signaling, and that will be, I probably will get through that today and may get started on the metabolic control, which really relates to energy, and that's some of the delta G stuff that Anna's been working about in the recitation. And so I will get to that. The material for the exam will cover everything through tomorrow. So we've got quite a bit of territory to cover. The exam, of course, will be on Monday. And I haven't decided if we will have only exam on Monday or if we will have a little bit of a lecture followed by exam. It kind of depends, again, on how far I get. So far, I'm in pretty good shape, but we'll see how that goes as I progress along today. So I'm already losing connections here, so this is fun. Okay. So signaling is important. When we think about multicellular organisms, we want all of the oars to be pulling in the same direction. We want the liver doing what the liver has to do. We want the bone doing what the bone has to do. We want the muscles doing what the muscles have to do. And this takes a certain amount of coordination. And the coordination that happens in a multicellular organism is essentially that of hormone signaling. All right? So I'm going to be talking today about a variety of different hormones and how they exert their cellular effects and essentially how they do, in fact, coordinate the body's actions. Okay? So these are going to be important for us to understand as we think about the way that a multicellular organism essentially has to function. So that's a very important consideration. Hormones are substances that are released by one tissue of the body, and they travel through the body to target tissues to exert their effects. So just like an email message gets released from one place, it travels through this vague black box we know as the internet, and it arrives at its destination where it elicits a response from the other end. So too do hormones get released, travel mostly in the bloodstream, and arrive at their destination where they bind to a target cell, and they bind to a specific receptor on a target cell and exert their effects. A lot of what I'll have to say involves the binding of the hormone to that receptor. Okay? That's critical. It's the first step in a process we refer to as signal transduction. So signal transduction actually relates to how information outside of a cell is communicated into a cell. Okay? So we want to understand how information that's outside of a cell gets communicated into a cell. And we will see a couple mechanisms by which these hormones exert effects inside the cell. There are many different ways in which they can. But the ones that we're going to talk about today involve alteration of enzymatic activity and alteration of transcription. Okay? Because it's through transcription that RNA is made and ultimately proteins are made. And so when we're talking about cells, for example, getting a stimulus to divide, ultimately that information is going to have to go to the nucleus, and that information is going to have to exert its effect on the chromosomes. Whereas if we're talking about an effect where an enzyme's activity has to be changed, then that information can alter an enzyme without having to travel to the nucleus and do all of these sorts of things. Okay? So that's important to keep in mind as I'm talking about things. One of the things that always strikes first-time people when they see action of hormones is the complexity of the signaling process. Okay? The signaling process really is phenomenally complicated in terms of the number of steps that have to go through. And again, I think the reference to the internet is an appropriate one. When we send out that message, and like I say, it goes to a black box, in fact, it goes to, if we have a wireless at home, it's going to our receiver that is grabbing that signal that's connected to basically like a cable modem. It's going to another computer from your house to a central computer. That central computer is linked to a network of other computers. So along the way, your message is traveling from one system to another system to another system. And we can think of each of those systems as being nodes, okay? From A to B to C to E to F to G to H, okay? And it might take a hop from quite a few computers to get from your computer to the person that you're sending the message to. And in that sense, the signaling system that exists in cells is not at all unlike that. So let's take a look at those systems here. These are some examples of systems, all three of which I will talk about today. Epinephrine is a system that also is, epinephrine itself is also called adrenaline. And it's involved in very, very rapid responses of organisms, frequently to danger. Okay, so we hear the stories about the person that's, sees the baby underneath the car, and they can go and they can literally grab the car, pick it up, and move the car, and they can. All right, and they can because of the physiological effects that are exerted as a result of action of epinephrine. Okay, so I'm going to say a few words about that today. Okay. Insulin is a hormone that we hear a lot about. We don't always think about it in terms of being a hormone, but that's exactly what it is. It's a peptide hormone. And it's a hormone that is released by our pancreas, and it's released to help the body deal with the problem of glucose. And what I hope to convince you by the end of today is that glucose is a poison. Glucose is a poison, and insulin is helping your body to deal with that poison. It's a necessary poison, but it's nonetheless a poison that causes problems for cells. The third hormone I'll talk about is one that is more intimately involved in the decision to divide or not divide inside of cells, and it's that of epidermal growth factor. And as its name would suggest, it is a growth factor. It is, in fact, a, God dang it. There we go. It is when it binds to the cell surface, it ultimately stimulates a cell's decision to divide or not divide. Okay, in this case, it's stimulating it to divide. All right. This thing keeps going that I will just turn this thing off. All right. So that's the three things that I'll be talking about today. Now, one of the things I've tried to impress on you so far is I've been talking about the control nature of a cell is that cells have to have ways to turn signals on, and cells also have to have ways of turning signals off. And as we will see, that will be a very important consideration, and I'll give you what I think will be a fun example of that as we get going a little further along. Okay. In fact, this simple figure here shows a little bit about the movement of that information through the body. We have a signal that's released in one part of the body. It goes to its target, which is a receptor on a target cell. If a cell doesn't have a receptor, of course, it won't respond to that hormone. And so not all hormones activate or affect all cells. So cells, by making or not making a receptor, become sensitive or insensitive to a hormone as it's released. That reception, which is what happens when the hormone binds to the cell surface, causes a signal to be moved across the cell membrane. And that signal being moved across the cell membrane results in an amplification. You might have one hormone molecule binding to a cell surface receptor, but you want to make sure that that one hormone binding actually causes big changes in that cell, because sometimes you're really completely reversing the direction that a cell is proceeding in, and that requires an amplified signal. The transduction is part of that amplification. That is, transduction meaning it's moving across the cell membrane barrier. And inside the cell, the response. So that response, like I said, might be activation or inactivation of an enzyme. It might be affecting the transcription of specific genes, etc. And then, most importantly, or I shouldn't say most importantly, but also importantly, are these blue lines going back. The cell is getting feedback, and the body is getting feedback about these processes so that it knows when to turn these signals off. All right. So a real good example, I've mentioned it once in class already, that of breaking down, for example, glycogen. Glycogen is a storage carbohydrate that we have. Enzymes are very good at breaking it down, and we don't want to let them run willy-nilly because we don't want to take all of our glycogen and destroy it. So we want to make sure that we're breaking down glycogen only to the extent that we need to and not any further, because that could ultimately cause some big problems. All right. Well, the next slide here that I have for you is that of a collective group of molecules, of which I'll be talking about actually all of them today, and they're called second messengers. So I need to define not only what a second messenger is, but also what a first messenger is. So when we think about the old days of the Pony Express, you had a person who had a letter that they wanted, Gesundheit, there's a silent sneeze. The person has a letter they want to mail in Kansas City to somebody in Denver, and of course they don't just dump it into a truck and it takes off. They've got ponies to do it. So they pass off that letter to the first Pony Express rider, who drives it to the second Pony Express rider, who drives it to the third, and probably by the time they got to Denver, it was probably about 20 or 30 riders that eventually got that information out there. Each of those people in that chain that are carrying the message is a messenger. All right? So the first messenger is that first carrier of the message, and in the body, the first carrier of the message is the hormone. So the hormone is the first messenger. I showed you 3 earlier. I showed you epinephrine, I showed you insulin, and I also showed you epidermal growth factor. All 3 of those are first messengers. All 3 of those are hormones. They are released to start a response. Now, second messenger is not quite so clear, although it's not overly complicated. The second messenger is a molecule that is produced inside of the cell in response to the message outside the cell. So, for example, if I release epinephrine and it goes and binds to an appropriate receptor on a cell surface, a series of events will happen inside the cell that will produce cyclic AMP. Cyclic AMP is the second messenger, because cyclic AMP will now act inside of the cell to exert the effects necessary for that cell to act. Okay? So first messenger epinephrine, second messenger cyclic AMP. I won't talk about cyclic GMP today. I didn't notice that was on there. It's related to cyclic AMP in that it has a guanine up here instead of an adenosine. Cyclic GMP I will talk about probably on the last, actually no, I talk about that, Gary will talk about that next term when he talks about vision. Cyclic GMP is a very important second messenger in vision, but I won't be talking about it here. Cyclic AMP I will talk a lot about, however. And so cyclic AMP is a second messenger. In every case, the molecules that you see up here on the screen are messengers that are acting inside of a target cell. So second messengers are always inside of target cells. Okay. Well, there we go again. All right, I think I'm going to bag this for right now. And just simply go back and talk through here. So, before I talk about hormones, let me get my laser pointer so I have something to, oh, wow, sorry about that. Clumsy professor syndrome here. All right, maybe something that's more effective than that stupid thing, stupid network connection that we've got. We talk about, we need to understand something about these receptors. So the receptors are, in every case, proteins that are in the membrane of target cells. So first hormone goes out, it's going to bind to a receptor. The receptor that it binds to is located in the membrane of a target cell. Okay? Now, when we look at the structure of these receptors, we see that many times they have a very common general structure. Okay? I'm going to show you that structure in just a minute, but suffice it to say that all of these processes right here are mediated by hormone receptors that are very similar in general structure. Okay? So it's quite a wide variety. Okay? all these processes, and no, you don't need to memorize those processes, but it's a very wide variety of processes, are ultimately controlled as a result of these receptors in the surface, on the membrane of these individual target cells. Okay, so I said that they had a common structure. What does it mean to say they have a common structure? In fact, what it means is that it looks something like this. So, schematically, I can draw the structure of a common receptor. By the way, as the name implied on the last slide, they're called 7TM receptors. The reason they're called 7TM is 7 is the number 7. TM stands for transmembrane domains. So what common feature that each of these receptors has is that they have 7 segments of their sequence that go from the outside, inside, inside, outside, outside, inside, inside, outside. And as they're crossing the membrane 7 times. Crossing the membrane 7 times. So that's the 7 transmembrane domains. Now, they aren't identical in structure. I don't want to claim that they're all identical. But they all do have that common feature of crossing the membrane 7 times. Okay? We're going to see 7 TMs coming up in a variety of ways and places. And they are interesting as a family of proteins. Okay? This is probably a slightly more accurate way of depicting them. But nonetheless, we have 7 of these guys that are crossing the membrane 7 times. This tries to give a little bit more accurate of a 3-dimensional image. We can imagine the membrane going outwards from here. Now, without these proteins on the cell surface, we have no way of, in fact, getting that signal from the first messenger inwards. So these guys play a critical role in this process of getting the message inwards. I talked about epinephrine. That's the first hormone. That is the first one I'll be talking about today. It is, in fact, derived from the amino acid tyrosine, and it's related to tyrosine. It's not a difficult process to make this from tyrosine. And I will say probably more about that later. Okay, well, let's talk about a signaling pathway that is a very, very common signaling pathway we see in cells. This pathway that you see on the screen, you will see later next term, for example, when you talk about how it is that fat breakdown is controlled by hormones. You will see it when you talk in a related way about vision and how vision actually happens. And now we're seeing it how this process occurs for dealing with epinephrine. Okay? So, what is involved in this process? I'm going to take you through step by step by step what happens in this process. And I will be honest with you that a lot of signaling involves this, goes to this, goes to this, goes to this, goes to this. And do I need to know those steps? The answer is in general, yes. Okay? Because we need to know what's happening in that process so that we can ultimately understand how or what happens with that signal. So I'm going to take you through the steps of this. All right. Well, let's imagine that I'm out and I go walking down the street and there's a grizzly bear that comes along, and I all of a sudden decide I've got to get away from this damn grizzly bear. All right. Well, my brain sends a signal that ultimately makes it to my adrenal glands, which says, hey, he's seen a grizzly bear, we've got to get out of here, we've got to do something about this. And what it does is it releases the hormone epinephrine. Okay? Epinephrine pops out into the bloodstream. Now, epinephrine travels by way of the bloodstream. It's not an immediate thing, but your blood flows reasonably fast, and you can get this signal to start getting it to target tissues literally in seconds. So it happens reasonably fast. Not instantaneous, but it happens reasonably fast. Okay? Epinephrine goes out and it bumps into cells. If cells have a receptor, it initiates this process that you see right here. If it doesn't have a receptor, of course, nothing will happen to that target cell. There are 2 types of cells I'm going to talk a lot about in this class, and they are liver cells and they are muscle cells. Okay? Liver cells and muscle cells play a very important role in this process I'm getting ready to describe to you. So epinephrine hits, let's say, a liver or a muscle cell, and of course that makes a lot of sense. Muscle cells, I need to get away. Liver cells, as we will see, are important sources of glucose. So they're going to provide energy for me to get away. or lift the car, or whatever it is I need. Epinephrine binds, and epinephrine causes this signal to cross the membrane of the cell. How does it do that, and what happens? Well, what it does is the binding of epinephrine, and you can actually see that little yellow epinephrine right there. The binding of epinephrine to the receptor causes the receptor, and you've heard this how many times now, this term, to undergo a slight shape change. Very slight shape change. Okay. On the inside of the cell, epinephrine, on the inside of the cell, the receptor, now this isn't working. This is really not my day, is it? On the inside of the cell, the receptor is interacting with a special protein called a G protein. We'll see why it's called a G protein in a second. The G protein has 3 subunits. I'll slow down since everybody's writing furiously. Okay, 3 subunits. It has an alpha subunit, which will be the main one that we're interested in. It has a beta subunit, and it has a gamma subunit. Okay? The interaction, or the change in shape of the receptor, causes its interaction with the G protein to change. Epinephrine is bound, receptor has changed, and now the interaction between the G protein and the receptor has changed. The interaction change between the receptor and the G protein causes the following to happen. Pay attention because people screw this up. It causes GDP to be removed from the G protein and replaced by GTP. Notice I said it was removed and replaced by GTP. I did not say GDP was converted into GTP. That is not what happens. GDP is removed and replaced by GTP. That causes a shape change in the alpha subunit, which causes it to release the beta and gamma subunits. Okay? So the alpha subunit is changed in shape. The beta and gamma subunits no longer bind to it. And the beta and gamma subunits were covering up a portion of the alpha subunit that it interacts with another enzyme. So now that that portion that it was being covered up, it's now able to bind to this other enzyme. The other enzyme is also located in the membrane, and it's called adenylate cyclase. Okay? Adenylate cyclase. So what's happened? Epinephrine's bound, receptor got changed, interaction with G protein got changed, GDP got replaced by GTP, beta and gamma subunits went bye-bye, alpha subunit interacted with adenylate cyclase. These are the steps that we've seen so far in this signaling process. The interaction of the alpha subunit with adenylate cyclase activates the enzyme. Adenylate cyclase becomes active, and adenylate cyclase catalyzes the formation of the second messenger. We had quite a few steps before we even got to this second messenger. Adenylate cyclase catalyzes the formation of cyclic AMP from ATP. Cyclic AMP, as you have seen previously, binds to protein kinase A. It binds to the regulatory units subunits of protein kinase A, causes them to leave the enzyme and activate the catalytic subunits. So the catalytic subunits are active, and now protein kinase A is ready to go phosphorylate target proteins. Wow, a lot of steps. This happens very, very rapidly. Once epinephrine has bound, this happens virtually instantaneously. Okay? So even though we're talking about a bunch of steps happening, these steps are not happening slowly. This happens very rapidly. Well, what have we done? We've gone through all this, and the one thing we have done is we have activated protein kinase A. Protein kinase A is capable of putting phosphates onto other proteins. That's what its name says, protein kinase A. Puts phosphates onto proteins. Now I'm going to briefly describe one set of proteins that this guy puts phosphates onto and explain the effect that it has. We'll talk more about it later next week. All right? Protein kinase A will start what's called a phosphorylation cascade, meaning it will phosphorylate a protein that will activate it, and that activated protein will phosphorylate another protein that activates it. Okay? Ultimately, we end up activating an enzyme that breaks down glycogen. Ultimately, we activate an enzyme that breaks down glycogen. The breakdown of glycogen gives glucose. It actually gives glucose 1-phosphate, but that's close enough. So let's review and think about what this signaling process has done. I'm scared. My adrenal glands release epinephrine, which now travel to target cells. If this is a cell in my muscle or in my liver, the beta receptors are there. They bind epinephrine. They activate the G protein by putting GTP onto it. GTP bound to the G protein is interacting with adenylate cyclase that cause adenylate cyclase to make cyclic AMP. Cyclic AMP activates protein kinase A, and a few steps later, glycogen gets broken down. The net effect of this process has caused the glucose level of this cell to increase. If this cell is a muscle cell, great. The muscle cell can use glucose directly, and it's got energy. I can use my muscles, I can escape, I can get away, and it's happened very, very rapidly. It's happened rapidly enough for me to escape, hopefully, the grizzly bear that's chasing me. If this, excuse me, if this is a muscle cell, then, I'm sorry, if this is a liver cell, then the liver cell doesn't have an awful lot of use for that glucose. What does it do? It dumps it into the bloodstream, and blood glucose levels go high so that it can travel to my muscles, and my muscles have more energy. So in both cases, I've released glucose so that my body has more energy and can get away and hopefully escape the danger that it thinks it's in. That clear with everybody? Okay. Now, I said that cells have to have a way of turning the process on, and they also have to have a way of turning processes off. We don't want this to be on all the time, because if we leave it on for very long, what's going to happen to our glycogen? Bye-bye glycogen, right? We're not going to get any good out of that. In fact, we're going to make a lot of glucose, which is, I've already sort of alluded to the fact, is poisonous. Okay? So we have to be careful we don't release too much glucose that we can't use. Well, fortunately, this process has a mechanism that turns itself off, at least part of itself. Okay? How does it turn itself off? Well, the answer lies in the G protein itself. So far, what you've seen of the G protein is that the G protein binds to GTP and it activates adenylate cyclase. If I leave that G protein with GTP in it all the time and I don't do something about it, what's going to happen to the production of cyclic AMP? It's going to be going on all the time, right? I'm going to break down. I'm not going to be able to stop that process, so I want to have a way to stop this process from happening. Well, the way I do it is it turns out that G proteins are actually very inefficient enzymes. They're very inefficient enzymes, all right? That's key to how they work. If they were very efficient enzymes, we would never get a signal. Well, now I need to tell you what it is they catalyze. They catalyze the breakdown of GTP to GDP. If they were efficient, as soon as they bound to GTP like any other enzyme, what would happen? They'd break it down to GDP, and when it binds to GDP, what happens? Beta and gamma subunits bind to it, and the signal is gone. However, it's an inefficient enzyme. This may take on the order of seconds to minutes for this reaction to get catalyzed. In the meantime, it's sitting here stimulating the production of cyclic AMP. Once the G protein converts GTP to GDP, it basically turns itself off. Because in the GDP form, it binds to this. This is what would interact with there. Now it can no longer interact with adenylate cyclase, and the enzyme is turned off. No more cyclic AMP is being produced. Does that make sense? Siobhan?
So I got confused. You
Okay.
went through at the beginning and you said that,
Yeah.
that the GDP is removed and replaced
By GTP.
by GTP.
Mhm.
And now you're saying that it gets converted to.
So that's right. So in this step right here, where the first hormone, the first messenger binds, GDP is replaced by GTP. That's what causes this to go over here. Okay, but this is like a ticking time bomb because this guy slowly is converted back to GDP. So in the order of seconds or minutes, this guy will convert itself to GDP and it'll essentially be back at this state. Okay. So we replace GDP with GTP, but we break down GTP back to GDP.
So it catalyzes itself back.
It basically catalyzes the conversion of GTP back to GDP and then hangs on to the GDP. All right? So that's how it ends up back in this state right here. This state is inactive. This state is active. So basically, the G protein is inactivating itself. That's an important theme we'll see later when we talk about an oncogene that does the very similar thing. Does that make sense? Did I answer your question?
Kind of, yeah. I'll have to think about it.
Okay. Other questions? Everybody understand what I'm talking about? Okay. So it turns itself off. Now, I've got a few minutes, and this is one that students always like, so I will take a minute and digress here. Well, I said the cells are control freaks, right? They want to be able to control every aspect of things. And I've just showed you how they turned off the production of cyclic AMP. But, uh-oh, what about all that cyclic AMP that I already made? It's floating around in the cell, right? Isn't that going to wreak havoc? Isn't it going to continue the signal going on? Well, the answer is, if there weren't another enzyme, yes, it would. All right? But the cell has another enzyme floating around inside of it that is almost always breaking down cyclic AMP. All right? So cyclic AMP, when we start making it, it rises, but it starts falling fairly quickly because this other enzyme starts breaking down cyclic AMP. What's the other enzyme? It's an enzyme known as phosphodiesterase. Phosphodiesterase. Okay? Now, why do I tell you this? Well, phosphodiesterase converts cyclic AMP to AMP, and therefore protein kinase A isn't stimulated anymore. Therefore, glycogen isn't being broken down anymore, and we've stopped the process in its tracks. That's good. Okay? Now, the interesting side of this is that there's a very common component of our diet that actually inhibits phosphodiesterase. It's in at least 2 cups in this room. It's caffeine. Okay? One of the ways in which caffeine works is caffeine inhibits phosphodiesterase. If we think about the inhibition of phosphodiesterase, what happens? Well, when phosphodiesterase is inhibited, concentrations of cyclic AMP don't fall, they stay high. And what happens when cyclic AMP concentrations are high? Your liver starts dumping out glucose in the bloodstream. You get a buzz from your coffee in the morning? Here's part of the reason why you get your buzz. Your blood glucose levels are going voing. They're going double voing if you're getting it in the form of a Coca-Cola full of sugar, or if you're getting one of those people, or you're one of those people who go to the coffee shop and you get basically chocolate milk with caffeine dumped in it and sugar and whipped cream, whatever they call those things, right? Okay? The reason you get that buzz partly is due to the molecular basis of what I've just showed you here in the signaling process. Is that a question? Oh, okay. Is that a question? I'm getting all kinds of imaginary questions. I see hands sort of going up. Okay, there's a question.
So, what did you say that it broke cyclic AMP down to?
It breaks cyclic AMP down to AMP.
AMP.
And that doesn't do any signaling here. Okay? Okay, so we get our buzz because we're inhibiting this process. Oh, you stupid thing. I'm going to turn this thing just to the off position, and it won't do that anymore. Okay. All right. All right, so that's kind of a cool thing. We'll talk more about sugar in the body when I talk about both glycolysis and glycogen metabolism. Okay. Well, there's a structural consideration of the G protein. As I said, it has 3 subunits. There's the alpha subunit there. There's where the GDP is bound in sort of the center of it. And there's the beta and gamma subunits that are color-coded on the side. The replacement of GDP by GTP does cause the alpha subunit to change shape. And in changing shape, it causes the beta and gamma subunits to be released, as I noted. That's a schematic representation of which you've already seen on the screen. In this case, it's bound with GDP. And of course, that's in the inactive state so that it can't activate adenylate cyclase. Adenylate cyclase is a transmembrane protein. You'll notice it's not a 7-TM. It's got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. You don't need to know that. But it's something different than a 7-TM. Virtually every protein that exists in membranes has transmembrane domains. It's just a question of how many it has and the actual sequence of those. The catalytic activity is inside of the cell. That makes sense because, of course, that's where the cyclic AMP is made and released. And that C there is actually, that's the C terminus, but I believe that is actually right there, the catalytic subunit as well. Okay. In words on the screen, that's the signaling process that I just described to you. Epinephrine binds to the receptor, activates it. GTP is swapped for GDP. This activates the G protein. Okay. The G protein goes to adenylate cyclase to activate it. That makes cyclic AMP. This is an amplification because this guy can make a lot of cyclic AMP. Cyclic AMP can go activate protein kinase, and protein kinase can do a variety of things. We're going to see protein kinase does a lot of things. I've only described one of them to you so far. Yes, sir?
So know the whole process.
What's that?
So know the whole process.
Yes, definitely. Yeah. Okay. Now, the example I've just given you shows the activation of an enzyme. The main enzyme here is protein kinase A. We will see that protein kinase A can activate and inactivate a whole bunch of other enzymes. So we can have a whole bunch of different things happening as a result of action of protein kinase A. All right? Okay, so our signal has been communicated. Another factor that comes up with respect to, actually, I guess this figure is showing the how the process is turned off. I sort of told you in words already. There's the GTP. The G protein is an inefficient enzyme, so it breaks down that GTP to GDP. When that happens, it no longer interacts there, and it binds to the beta and gamma, and therefore this thing doesn't cause adenylate cyclase to make cyclic AMP any longer. So as a consequence, that process, that signaling process has been completely turned off. Another thing that cells have as sort of a protector is they want to make sure that this signal really does, in fact, get turned off. What happens if epinephrine gets in the middle of that receptor and it gets stuck there and it leaves the receptor stuck in the on state? That could be bad, because if it's stuck in the on state, what's going to happen? Well, it's going to be continually stimulating a G protein to be active, which is going to continually cause adenylate cyclase to continually make cyclic AMP, to continually activate protein kinase A, to continually break down glycogen, and eventually the cell burns itself out. So there's a protection built into the system for basically stopping that from happening, and it's shown here. There's 2 ways of stopping this signal. One is that epinephrine can just come back out. If it gets stuck, however, and doesn't make its way back out, the cell can actually recognize that, and don't ask me how it does that, by action of an enzyme known as a receptor kinase. What the receptor kinase will do is it will phosphorylate a portion of that beta-adrenergic receptor, the receptor for epinephrine. Okay? The receptor for epinephrine, I don't know if I said that or not, it's called the beta-adrenergic receptor. That's what it's called. Okay? So it phosphorylates a portion of the beta-adrenergic receptor. You see that happening here. And that phosphorylated receptor is a target for an enzyme called beta-arrestin. Beta-arrestin binds to those phosphates, stops the G protein from getting activated, and basically turns this whole system off. Okay? So if there's a problem, this is a very important mechanism for preventing that problem from continuing. Okay. This makes sense? Okay. So 2 ways of stopping or turning off the beta-adrenergic receptor. Okay. Let's see, where was I? Let's talk about another system, another signaling system. And this one is actually a little bit less specific than the one I've just described to you. I'm going to lead it to activation of an enzyme and then sort of leave it hanging there. Okay? This signaling system involves a different 7-TM, and it involves a different kind of a messenger. All right? So I'm not going to talk about the first messenger. I'm not going to talk, in fact, about how these molecules are made. All I want to do is talk about what happens after the messenger has bound. So the first messenger binds, and it stimulates the activation of an enzyme in the membrane called phospholipase C. So we've had a first messenger bind to a receptor. We've had the receptor activate an enzyme in the membrane called phospholipase C. Okay? Now, phospholipase C, its name actually tells us what it does. A lipase is something that cleaves a lipid. Phospho says it's a phospholipid. So this enzyme cleaves phospholipids. Well, where do we find phospholipids? They are abundant in the membrane. That's what makes up the lipid bilayer. What this mechanistic process is doing is it causes activation of phospholipase C, which now acts on some of these molecules that are in the membrane. Here's one of them, phosphatidyl inositol 4,5-bisphosphate, or as you're more likely to call it, PIP2. PIP2 is a substrate for phospholipase C, and phospholipase C cleaves PIP2 into 2 second messengers. There are 2 second messengers that it makes out of this. It clips this bond right over here, and when it clips this bond, it releases IP3, and it releases the other half of it, known as diacylglycerol. Now, what are the fates of these two? Diacylglycerol remains in the membrane. We'll see it becoming important in a minute. IP3 is released to go into the cytoplasm, and that's where it goes. Okay? Now, both of these molecules are going to act as second messengers in the process I'm getting ready to show you. But are we clear that phospholipase C is clipping this guy here that's in the membrane and releasing 2 second messengers? Okay. This now shows us what's happening as a result of what I've just described to you. Okay? Here was our PIP2. Phospholipase C got activated, and it cleaved PIP2 into IP3 and into diacylglycerol. That's the DAG that you see there. And yes, you can call it DAG if you wish. Okay? What happens? Well, for one thing, IP3 travels to the endoplasmic reticulum. If this is a muscle cell, we might call it the sarcoplasmic reticulum. The important thing is that where it's traveling is to a cellular organelle that contains a lot of calcium. Cells sequester calcium into organelles. Cells are very careful about how much calcium they have present inside the cell at any given time, at least in the cytoplasm or nucleus. Why? Because calcium will cause DNA to precipitate. So cells, when they use calcium for a lot of things, they tend to sequester it. So they've got it sequestered here in the endoplasmic reticulum. IP3 comes to this receptor on the endoplasmic reticulum, and it binds to it. And the effect of binding to that receptor on the endoplasmic reticulum is to release calcium. So now calcium comes streaming out of the endoplasmic reticulum. Yes, the concentration out here increases, not to deadly amounts, but it increases in the cytoplasm. The calcium travels up here to our target. Yeah, I heard that sigh. It travels to our target, known as protein kinase C, a different protein kinase. What does protein kinase C do? Well, protein kinase C is activated by two messengers. One of them is calcium, and the other one, there was our friend diacylglycerol. So the combination of these two cause protein kinase C to be activated, and as a consequence, protein kinase C can now go and start phosphorylating a bunch of proteins. This system is important in, among other things, modulating blood pressure. Okay? When we think of muscular contraction, one of the things that's very important for us to understand is that calcium is a signal for muscular contraction. This signal can help constrict, as appropriate, blood vessels and other things necessary using calcium ions as a signal. Okay? So as a consequence of this process, a physiological response is mounted, and it's mounted through the activation of protein kinase C. Okay, everybody's starting to look a little tired, so maybe we'll take a brief break and come back, and I will dive into some more more signaling pathways.
I have a question.
Yes, sir.
So why is it that people get more resistant to caffeine then?
Well, caffeine response is complicated. They don't get overly resistant to caffeine. They just get more, I guess, less responsive to it. But it's, there's more to the caffeine response than what I've shown you here. This is a part of the response, and you will release some calcium. There are also neurological effects that caffeine has. And like any other thing that happens inside the brain, you see changes happening with dose.
Yeah.
Yeah.
Because I know a lot of people who are like, no, one cup of coffee doesn't even do.
Yeah, and I'll tell you, your body's response to caffeine changes with age. What I drink in terms of coffee today, well, I'll get you on the camera. Just a second, sorry. Forget before I do this. That's my signal. That was the break where I know where I did that.